Good morning children and welcome to the online teaching session. Students, in the previous video, I have started a chapter genetic material and protein synthesis. Okay, in that chapter, I taught you the definition of genes, properties of genes, old and new concept of gene and its function. Okay, now after that, we have to start with the structure of DNA and to consider the DNA as a genetic material. Clear? Now children, from your syllabus, Griffith experiment and the experiment which was done by MacLeod, Avery and McCarthy has been omitted, at least for this year. Okay. So these two experiments are not given in your syllabus, and that's why we will directly start with the experiment given by Hershey and Chase. Okay. But let me give you the summary of these two experiments. What had actually happened in the beginning, the scientists, they all were very confused that what could be the genetic material which transferred from one generation to other generation. Okay. And for that, they did many experiments. Then, one scientist in 1909 came forward and he did experiment on mice. Okay. And this is called as the Griffith experiment. Although he did not reach to any conclusion, but at least he was the first scientist who said that something is there inside the body which transfer from one generation to the other generation. And his experiment was finalized by the three more scientists after 16 years. Okay. And these scientists told us that this DNA is the genetic material. But still it was not 100% confirmed. Then came two scientists named as Hershey and Chase. Okay. A.D. Hershey and N. Chase. Okay. Now, just to avoid the confusion that what would be the genetic material, whether it may be the protein or it may be the DNA. So, out of the two, at least one will be the genetic material which carry our genes and which transfer from one generation to the other generation. Clear? So, these two scientists came forward and they did one test. They did one experiment. For that experiment, they took two things. Clear? What they took? They took T2 phage virus, not bacteria, T2 phage virus. And then they took E. coli. Okay, this is one bacteria. Along with this, they took radioactive sulfur and radioactive phosphorus. Okay children, now see. Now one by one. They took T2 phages, that is they took virus. Why they took this virus? Because the body of the virus is like this. That is, the body of the virus is basically made up of only two things. One is protein and the next is DNA. Okay, I am drawing it. Now see. like this. Okay, let me make it shorter. This is protein capsid. Okay, and all these are tail fibers or tail pieces. Clear? Then this is your end plate. Let it be with this only. Let me mark it like this. This is the end plate. Okay. Protein capsid, tail piece and end plate. Now, this contains protein also. Circular protein. So, what we can write? This is protein. Then, this T2 phage virus contains DNA. Now, there is a perforation, children. Okay. Now, this is the structure of the T2 phage virus. Okay. Now, see. Virus is basically made up of two things. One is the protein and other one is the DNA. Now, out of this, it was easy for the scientist to say that which one is the genetic material. Now, see. Why they have taken this E. coli, and the reason is the virus always replicate inside the body of a bacteria. If there will not be bacteria, viruses will not be able to replicate. Okay, every time these virus needs a host body. This we have already studied in class 11. Now, two things are clear. Rest is, they have taken radioactive sulfur and radioactive phosphorus. Reason, this sulfur is the important component of protein. Okay? And this phosphorus is the important component of DNA. And they have taken this as radioactive so that these sulfur can be incorporated with protein and this phosphorus can be incorporated with DNA and they can be easily visible. That's why they have taken the radioactive sulfur and radioactive phosphorus. Okay, so these were their requirements. Now we will begin with the exercise. That is the experiment given by them. Now see, we know very well, children, that if a virus sits on the E. coli, let me draw, if a virus like this, okay, if a virus sits on the E. coli, then it does 4 steps. Number 1, adsorption. Okay. After that, injection, and after injection, replication, and then lysis. Now see, what is the meaning of all these terms? Suppose if a virus sits on the surface of a bacteria. Okay, we know very well that on the surface, the term is called as adsorption. This is not absorption. If something sits on the surface, adsorption. Adsorption is a surface phenomenon. Clear? Now what happens? The virus sits on the surface of the bacteria as soon as it sits, it removes all its end plates and try to penetrate inside the plasma membrane. And the genetic material that is already present inside, it will move here through this perforation that I have drawn in the end plate. Okay, now this is the end plate, this is the perforation, and the genetic material will be injected. Okay, so two steps are clear. Number one, the virus will sit on the surface of the bacteria. Okay, it will absorb all its end plates and everything. It will sit and penetrate, and through this hole, the genetic material will be injected. Two steps are okay. Now, inside, this genetic material will replicate. Okay, and it will make the virus like this. After this, these viruses, they will, with the help of enzyme that we will see afterwards, they will break down this membrane and they all will come out. So this has to be done by the viruses. Clear? Now what these scientists have done, they have cultured the T2 phage and E. coli together in the medium of sulfur as well as in the medium of phosphorus. See what they have done. Now I am drawing it like this. Okay, what they have done, they culture, this is the common step. What they culture? T2 phase plus E. coli in sulfur medium. Now how was the sulfur medium? It was radioactive sulfur medium. The same step, they culture T2 phases plus E. coli in phosphorus medium. This phosphorus was also active. Okay. Now, this is your first and this is your second experiment. After doing all these things, they put everything in a test tube. Okay. And they shook the test tube very well. Okay. They shook it, or we can say that they centrifuged it. Centrifuge it. Here also we can write down centrifuge it. Now children, see what happens. Being the E. coli heavier, it will come to downside. Okay, now see, we all know very well that adsorption had already taken place. Now, if we will put them into the test tube and if we will shake the test tube, then being lighter, the T2 phase, the capsid of the T2 phase, it will remove from the surface of the bacteria. Okay? Because it has already inserted the DNA material inside the body of the E. coli and it is loosely attached on the surface of the bacteria. So, as soon as we will shake them, the capsid will remove. Clear? So, at the upper side, that is in the supranatant part, we can see the capsid, and in the palate part, we can see the E. coli. Now see, same result. Here, this is the supranatant part. And the capsid is here only. Clear? Now this is, here I am writing, this is the supranatant and this is the palate part. Now see children, what had happened now? They found that the radioactive sulfur was here only. Okay? Radioactive sulfur. It was here in the supranatant part. It means the protein did not go inside the bacteria. Because we all know that this sulfur is an important component of protein. Okay, it is an important component of protein, and as they found sulfur here only, so they observed that or concluded that the protein did not go to the bacteria. Now, see what had happened in the second. Here in the palate part, they found radioactive phosphorus. Okay, and they concluded that this phosphorus had been entered into the bacteria. It means the phosphorus is an important constituent of DNA. So we can say that DNA has been transferred to the bacteria. Okay, and then only bacteria replicated, or the virus replicated and replicates the bacteria, and the process occur. Now, this proves that, what is the result? This proves that DNA is the genetic material, okay, which transmit from one generation to the other generation. Okay, it carries all the information and then the transfer from one generation to the other generation. So children, this is the experiment of Hershey's and Chase, and they proved that DNA is the genetic material. Why DNA is the genetic material? Because DNA is the, this phosphorus is the main component of DNA, and the phosphorus is showing that it is the genetic material as it has been transmitted to the bacteria. Clear? So this is all about the experiment of Hershey's and Chase. Now, in the next part, we will see the structure of DNA that was given by the different scientists. Thank you, children.